Heart disease is the #1 cause for death in humans. Despite the proven efficacy and general safety of standard of care drugs like statins, the development of safer and more effective treatments to prevent heart disease remains a key priority for public health officials. Multiple approved therapies for controlling heart disease are on the market. The proven efficacy of these interventions, such as lipid-lowering drugs like statins, creates a major hurdle for further development of new therapeutics that need to demonstrate competitive advantages over existing standards of care. Furthermore, there is an increasing concern amongst leading scientists and regulatory bodies that changes in circulating lipids, such as good cholesterol or HDL, and plaque size following therapeutic interventions are not the most informative surrogates for the changes in the risk of a heart attack or stroke in patients. The evaluation of the effect of novel therapies, genetic factors, and lifestyle changes, including diet and exercise, on cardiovascular, or CV, outcomes is a major clinical challenge. These CV risk endpoints change over a very long period of time, from years to decades, and requires costly clinical trials to discern a significant benefit from treatment. Recent high-profile cases have implicated approved compounds for a variety of indications such as pain relief and type 2 diabetes to statistically increase cardiovascular morbidity and mortality. Any indications of CV risk may not be observed until after a novel therapeutic has been taken into late-stage human trials or even approved. New technologies to enable prospective evaluation of the efficacy and cardiovascular safety of new therapeutics are desperately needed when evaluating cardiovascular disease with the comorbidities of other diseases that may affect circulating lipids and atherosclerosis. The Entellus Cardiovascular PhysioLab platform provides a means of predicting changes in CV risk in a cheaper, faster manner than clinical CV outcomes trials. As shown graphically in the lower part of the slide, the CV platform contains a representation of the key biological pathways and dynamic feedbacks that drive the development, progression, and morbidity/mortality associated with cardiovascular disease, including cholesterol metabolism, platelet activation and aggregation, systemic and local vessel inflammation, atherosclerotic plaque formation, and endothelial cell dysfunction. Changes in these factors, when integrated together, lead to changes in patient plaque stability and ultimately changes in the likelihood of a CV event. The current CV platform focuses on coronary heart disease, or CHD, endpoints, but internal efforts are underway to extend the platform capabilities to the prediction of stroke. As indicated on the left, the platform contains a diverse set of virtual patients that embody the diversity in the actual cardiovascular patient population, including patients with different risk factors for cardiovascular disease, such as diabetes, hypertension, rheumatoid arthritis, and smoking, circulating lipid compositions, patients who have recently had a heart attack, and patients with specific genetic factors that result in very high circulating lipid profiles. Virtual patient populations have been created to represent specific clinical populations with a diverse combination of risk factors, such as diabetes, hypertension, and smoking, as well as patients that are not typically recruited into clinical trials but that may be present in the post-approval population. Changes in a number of clinical outputs relevant to real-life cardiovascular disease, including circulating lipid profiles, particle size distribution, measures of plaque burden, endothelial dysfunction, and CHD risk can be predicted as a result of an intervention of interest, as shown in the middle. Finally, the right side of the slide shows that a number of approved therapeutics are represented in the platform, enabling evaluation of the efficacy and cardiovascular safety profile of novel interventions on a background of these approved therapeutics. The CV PhysioLab platform contains a diverse and complex biology and has been subjected to extensive validation against a number of clinical trial protocols for a variety of therapeutic interventions. Only a subset of the validation studies are shown here. For additional details, please contact Entellus. Predictions of the CV PhysioLab platform are consistent with various lipid, plaque, and CHD risk endpoints reported in clinical trials and meta-analyses of different types and potencies of statins, CETP inhibitors, ACAT inhibitors, and acute therapeutic infusions such as reconstituted HDL. Furthermore, the platform architecture is able to appropriately capture the diverse pathophysiology embodied in different types of cardiovascular patients, including mixed dyslipidemic, familial hypercholesterolemia, and acute coronary syndrome. Additional validation against more recent clinical trial data for different interventions such as cholesterol absorption inhibitors is continuously ongoing.